My name is Mitchell Hayes. I'm a urologic oncologist at Moffitt Cancer Center, and I was really lucky to receive the 2024 KCA Trailblazer Award for our work that primarily looks at immunoresistant clear cell renal cell carcinoma. So this project was sort of initiated after a pilot program that looked at some surgical samples from patients with clear cell renal cell carcinoma who had received immunotherapy but still had residual immunoresistant tumor left in their kidneys. And we primarily are interested in the tumor immune microenvironment, which is looks at not just cancer cells, but also other cells surrounding the cancer, including macrophages and fibroblasts and things that we know are very important to treatment response. And so we use a very high resolution camera to look at transcriptomics within the tumor immune microenvironment. And this project primarily was to test some hypotheses that arose from that pilot project, where we found that there was a gene that was spatially enriched sort of at the borders of the tumor. That gene was YES1, and also to look for autocorrelation, which is sort of like coupling of important genes that are related to the epithelial to mesenchymal transition pathway near the edges of tumor. So very excitingly, we were able to validate one of our findings that looked at YES1 and found that it was increased in expression in this expanded cohort of patients that we looked at. And we used a slightly different methodology, so we feel very comfortable that this validation is a real signal. And so then we turned around and turned it into a bulk RNA gene signature, which we tested in an external cohort of about 18 different institutions across the US, part of the Orion network. And we found that indeed the YES1 signature was associated with worse responses to immunotherapy in patients who develop metastatic disease. So this is a very exciting finding, and our next steps are to look at what are the potential biomarker implications or therapeutic vulnerability implications of these findings. So we have published two abstracts currently, and we have several others in submission. And this work was also, I think, very fundamental to sort of my own career trajectory in applying for additional funding that uses sort of some of the same methodologies, novel methodologies. And I was very lucky also to receive a 3-year DoD sort of Fellows Award, which looks at similar aspects of the tumor immune microenvironment, but in papillary renal cell carcinoma. So we've started that project and are making some headway there too. Well, I think that this award was sort of the first step in really becoming part of the KCA community, which is an amazing community of people, very friendly, amazingly accomplished researchers and advocates, and this is my third conference at the IKCS, but it's truly my favorite conference because of that sort of community aspect, and also the focus on mentorship. So I've met a lot of amazing people, and there's so much to learn from them, especially other researchers. And so this grant award was really kind of the first step in being introduced to that community. And also I think it was the first opportunity for me to become more independent with my research. So this award was kind of at a transition time for me from graduating from my SUO fellowship as becoming an independent urologic oncologist, and also a PI on a project. So my mentor, Brandon Manley, who has been extremely generous to me, as a fellow and now as a young faculty, was my mentor as a fellow. But, this award being granted to me as the PI has allowed me to kind of, start removing some training wheels and try things on my own and starting to kind of, learn some important skills in terms of, how to conduct a project and how to do things efficiently. I think that, one thing that I've had the opportunity to do in some follow-up meetings, related to this award, is speak with advocates, speak with, folks who are, donors and trying to see what excites people in the community, because, I think we can become a little myopic, especially on the kind of the bench side, and trying to understand exactly, how things sort of work on a mechanistic, sort of microscopic scale, and that is really essential and important to, the work that we do as a whole. And, the value of basic science obviously is creating that foundation for, what is to come in the future. But, there's definitely a sense of urgency too, with patients, and they want to, they want to see how this translational research is going to bring things to patients faster. And I think that's been a work in progress just for the biomedical research community over many decades. Is how do we speed up this process and how can we bring things to the bedside faster? But having some face time and being able to talk to people really makes you think about that at every step in the process. So we're doing all this technical work, but I'm constantly reminded of what is the end goal. The end goal is to help patients and to try and, for example, increase durable responses to immunotherapy. So I think that has been really important for me remembering that throughout the process. There are two aspects I think I'm really excited about. One is sort of the biomarker side of things, and then the other one is sort of the therapeutic side of things. So on the one hand, we've developed this gene signature that seems to have some predictive value in terms of, what patients receive for systemic therapy when they develop metastatic disease. Um, these samples that we use were primary samples, so most of these patients actually had surgery, and then when they developed metastatic disease later, then basically we were able to use the signatures from their original surgery to decide over what's the right, what was the response to therapy at the time of metastatic disease. So, we really hope that we can sort of validate, that sort of gene signature biomarker and whittle down exactly what are the features that is, making somebody not respond to immunotherapy. So that's sort of like, I think, something that is a lot more immediately, translatable in terms of, patients and there are many gene signatures that, are sort of, trying to do the same thing. And so I wouldn't be surprised if there was some overlap with some of the other existing ones. And then on the other side is sort of the therapeutic side. So yes, one is non-receptor tyrosine kinase. There are drugs that can target yes1 in addition to other proteins that are like that. And these drugs have actually been tested in other types of cancers like lung cancer. So some of the sort of future steps we would like to do would be to try and test some of these drugs in something like a mouse model and see if it synergizes with immunotherapy. So I think that there's two very exciting paths, and we are excited to kind of see what the next results would be from that. I think that one of the pains of being a researcher or being a doctor, being a patient is that as I mentioned, the road to progress is so long, and from initial discovery to actually having treatments that are making a difference. But that process has over the years gotten shorter and shorter. We've become better about sort of, projecting into the future. How do we, take this mechanism and, bring it to the bedside? What's the fastest route? And, there's a lot of, thought leaders in kidney cancer who have done this really well. People at other institutions that come to this meeting and kind of teach us how they did it. And so that shortening of that lag time is sort of finally coming into fruition now. And the great thing about sort of kidney cancer is that I think kidney cancer researchers are really good at looking at the advances made in other cancers that might be a little bit more common or better funded, and trying to apply them to kidney cancer. And it just seems like we're doing that more now than we've ever done before. And over the next decade, that's going to come into fruition. So, we've made a lot of progress, and especially with, the introduction of immunotherapy, and we're building on that momentum. So, I think that's, one thing to be excited is sort of that shortening lag time for the translational research. The other thing that I think is really exciting is, everything that's related to AI, not necessarily sort of the power of deep learning and sort of these, uninterpretable algorithms, but the way that it can actually help the researchers is I think a little bit less emphasized. Like, for example, these tools that are bringing research to us, search tools, and the ability for researchers to have access to other same person type research, that brings a lot of clarity to my own process and my own work, I think. And so those are a little bit of the unemphasized advantages, I think, of like AI and advanced search tools, and bringing sort of the research community together in a more coherent way. Not necessarily like all the deep learning algorithms, but bringing the people together who are going to actually do the work.